Today we are going to talk about how signals travel through our brain. These signals dictate our actions and personality. Signals travel through our brain through a process called action potentials, which occur in every neuron. In our brain, there are about 86 billion neurons.
The first step to understanding action potentials is understanding what exactly a neuron is. Neurons, also known as nerve cells, are one of the most important units of the brain and nervous system because they're responsible for sending messages between different areas of the brain. Specifically, most neurons are responsible for receiving sensory input from the world, for sending motor commands to muscles, and for transforming the electrical signals at every step in between. Neurons have a lot in common with other types of cells, but they are structurally and functionally unique. Neurons have three main parts: dendrites, soma, also known as cell body, and an axon. A dendrite is responsible for receiving information from other neurons. The soma is the cell body of a neuron and contains the nucleus and various other organelles. And the axon is where the electrical impulses from the neuron travel away to be received by other neurons. What is the resting membrane potential and how is it maintained? The resting membrane potential is the voltage across its membrane. This is determined by the concentration gradients of ions across the membrane and the membrane permeability to each type of ion. The resting membrane potential is negative 70 millivolts for a neuron cell. A pump actively transports potassium and sodium ions against their electrochemical gradients. Potassium moves inside the cell and sodium moves outside the cell. This allows the concentration gradient that these ions travel down to be maintained, and therefore the resting membrane potential to be maintained. The sodium-potassium pump moves 3 sodium ions out of the cell for every 2 potassium ions it moves inside the cell continuously. It therefore maintains the large potassium ion gradient across the membrane, which in turn provides the basis for the resting membrane potential.
Neurotransmitters and electrical potentials help to communicate between neurons by sending and receiving signals. The dendrites on a neuron specifically have transmitters on them, which receive the signals, then transfer them back to the cell body. The neurons become stimulated when the nervous system is triggered and action potential begins. This happens when hitting threshold, and membrane potential begins at negative 55 millivolts. The axon hillock is first to be stimulated and to trigger the action potential. Sodium and potassium ions line the cell membrane, trying to get open the channels and return back to the resting membrane potential. This is when the electrical potential across the membrane separates charge. An action potential is when the neuron is stimulated and it fires an electrical pulse in order to react to what is stimulating it and send a message to other neurons. A concentration gradient is used to show how our ions differ in concentration inside and outside the membrane. Potassium and sodium ions are flowing back and forth between membranes at different voltage levels through the channels. Voltage measures the amount of energy generated by separate charges and tells the membrane potential. When the voltage hits threshold, that is when action potential begins.
The threshold of a cell is when the inside of the cell membrane reaches negative 55 millivolts, which triggers the beginning of the action potential.
The membrane begins depolarizing, and positively charged ions begin to go into the cell body right away.
These positive ions are sodium ions that flow inside the cell when the threshold, negative 55 millivolts, is reached, which triggers the first voltage-gated sodium channel to open.
When depolarization happens, the membrane potential moves past zero and goes all the way to over 35 millivolts. Then, after the voltage-gated sodium channels inactivate, the positive charge flows back into the cell with the potassium gates open. During repolarization, the surge in potassium ions out of the cell lowers the voltage, so the membrane potential slowly starts moving back into the negatives.
During repolarization, the voltage-gated sodium channels close and the voltage-gated potassium channels open when the charge reaches positive 35 millivolts, allowing for the potassium ions to flow outside the cell and causing the cell membrane to move towards its resting membrane potential of negative 70 millivolts.
Hyperpolarization happens, which means the potential drops to lower than resting membrane potential, because so many positive ions move out of the cell. And then the membrane finally returns back to rest. The sodium and potassium pumps in ion channels help to even the gradient. Action potential varies by speed and conduction velocity.
This chain reaction of the opening and closing of potassium and sodium voltage-gated channels continues down the axon and creates the shape of the action potential. The reason that the action potential moves in one direction down the axon is because after the voltage gates open, they need to close and reset before they can open again. This is called the absolute refractory period, as an action potential cannot fire during the reset. Two factors determine how fast the action potential travels: the diameter of the axon and the myelin sheath that surrounds the outside of the axon. The larger axon diameter allows signals to move faster since there is more space for the ions to travel inside the cell. As more ions flood into the axon, they are less likely to bounce into other proteins or ions inside the cell and slow the action potential down. The myelin sheath also makes the action potential faster. Inside the axon, negative charges repel each other and get pushed to the side of the membrane. This attracts positive ions on the outside of the cell membrane and creates a thin layer of ions on the inside and outside. The myelin sheath creates a thick layer between the inside and outside of the cell, making the positive and negative attraction weaker. This causes the negative ions to be more spread out throughout the axon, so when the action potential comes rushing by, it's easier to depolarize because the ions are less concentrated. The spaces between the myelin sheaths are called nodes of Ranvier, where the voltage-gated ion channels are located. Since there is no sheath, positive ions gather outside the cell where the gates open, and more positive ions flow in. It is very important to understand how neurons fire because they define the functions of our bodies and help us understand how we work. For example, when our hand is over something that is hot, our nerves in our hand send signals to our brain that are processed and sent around by neurons and causes us to remove our hand from the source. Neurons dictate our personalities, our body movements and muscle movements, and our overall ability to sense things around us. Thank you so much for watching, and we hope that you learned important things about neurons, action potentials, and the way they work.